is we're going to talk about the structures of macromolecules. Of course, macro means big, molecule is a chain of atoms. Now, if we look here, there are really four building blocks of life. We have the amino acids, fatty acids, nucleic acids, and of course we have the saccharides, which is the carbohydrates. We know that the building blocks of the amino acids are going to be, actually the amino acids are the building blocks of proteins. We know that monosaccharides are the building blocks of carbohydrates. We know that nucleotides are the building block of DNA and RNA. And we know that fatty acids make a variety of structures such as phospholipids, fats in general, and also cholesterol. Anyways, there was some confusion about the nucleotide and its ability to form complex chains called DNA or RNA. And we're going to briefly touch upon this sugar here. And this is a sugar called either ribose or deoxyribose, depending on if it is a part of DNA or RNA. So we're going to pretend that this is DNA, just for fun. Now we know that DNA is built out of a five-sided sugar that we call either deoxyribose or ribose. We know that it's built around a chain of carbons and one oxygen atom. Now, if we look here, there's actually another carbon that is attached to a phosphate group. And the phosphate group is of significant importance here because that is what allows nucleotides to bind. And also for authenticity's sake, we're going to build a nitrogenous base. So that's the side group that I drew there. Now, we have a saying in genetics that DNA and RNA are always built what's called 5' to 3'. It'll make more sense as we talk more about things like DNA replication and also RNA synthesis. It will make more sense then, but for right now, we're just going to say that DNA binds 5' to 3'. What does that mean exactly? Each carbon in this chain is given a prime number, and it's going to look like this. It's going to look like 1, 2, 3, 4, and 5. We call these primes. Each carbon is given a number called a prime. And if we look here, the bottom left of the base has a 3, and the carbon that attaches to the phosphate has a 5. So when we say that DNA binds 5' to 3', what we really mean is that that's the direction that it binds. So I'm going to go ahead here and draw another five-sided sugar. So as you can see, what I've done is I've drawn another nucleotide. Recall a nucleotide is the building block of DNA. I've drawn that underneath the first one that I drew. Now, how are they going to connect? Well, keep in mind that I said that DNA always binds 5' to 3'. If we're going to draw the directions again, we have 1, 2, 3, 4, and 5. Those are all the carbons in the chain. So how's this going to work? Well, we see here that we have 5', we see here that we have 3', and we see here that we have a phosphate. In order to build a chain of nucleotides, we need to form what's called a phosphodiester bond, a very strong covalent bond. And this isn't 100% accurate, but basically what's going to happen is that this phosphate group is going to connect on to this carbon. So now we have a phosphodiester bond. Phosphodiester bonds are very strong bonds that link together nucleotides. Now, if we want to continue building upon our chain, we can build yet another nucleotide. Keep in mind that's only a half of one. We see here once again we have 5' to 3'. the phosphorus that is attached to the 5' carbon on this middle nucleotide is going to attach to the 3' carbon that's on this new molecule that I drew. Hence, we are building our DNA strand. We see this DNA strand is built by this nucleotide, 3' to 5' phosphodiester bonds. We have here 3' to 5' phosphodiester bonds. So what direction is this built? It's actually going to be built in this way from the bottom up. 5' to 3', new nucleotide, 5' to 3', new nucleotide. So the thing about DNA is directionality. It's going to go from the base, and it's going to be built that way. Hence, it's being built 5' to 3'. Now you're probably saying, well, what does that mean exactly? DNA is actually going to be a two-stranded molecule. Two strands connected at the nitrogenous base here using a type of bond called a hydrogen bond. And if we're going to look at this briefly, I don't want to go into it too much again, but the other strand is going to be called anti-parallel. We know that parallel means side by side, we mean anti means against. Similar to what I told you about DNA and RNA. So it's going to look like this. Again, not perfect, but we see the nucleotides actually are going in a different direction. Going to just illustrate what this would look like. And if we're going to do some directionality briefly, we have 5', 3', 5', 3', 5'. Oops, that's a 4. Not a 5. That's a 5. So the phosphodiester bonds will be built like this. Because it's always built 5' to 3'. So this strand would actually be built in this direction. And this strand here, which you probably have a little bit trouble seeing, but this strand here is going to be built in this direction. From 5' to 3', 5' to 3'. Recall that these are phosphoruses that are bound. So these make phosphodiester bonds. And that is our schematic of the DNA molecule. So today we're going to recap fatty acids, which I didn't really have a chance to talk about too much last class. But anyways, we see the beginning here of two chains, saturated and unsaturated. The way that they differ is more or less how occupied the carbon molecules are. We know that saturated means completely full. If you go to target and every check stand is full, we call that saturated. It's going to be an important term actually as we go through this course. Now, unsaturated means that not every carbon molecule is going to be saturated with another atom of importance called hydrogen. So if we look here, each chain is going to start off looking pretty similar. So I'm going to draw here the hydrogens. You can draw along too. So as you can see here, both molecules are looking pretty similar. They're both saturated with hydrogens and they each have 4 bonds. If you look here, carbons bonded to carbon, bonded to carbon, bonded to the hydrogen, bonded to hydrogen. That's 4. Now one thing to keep in mind is the AP is going to throw some problems at you, and I will too, where at the end of a molecule there's going to be a carboxyl group. Know that a carboxyl group is carbon, double bonded to oxygen, single bonded to OH. And any molecule that ends with acid is going to have a carboxyl group. Of course, we know that they're found on amino acids and now fatty acids, and sometimes they'll show you a nucleic acid. Now, as we see here with the saturated fatty acid, it's going to have a very linear appearance. However, unsaturated is going to change in one significant way. It's going to have periodic carbon-carbon double bonds. Now what's going to happen with this carbon-carbon double bond is it's only going to be able to bond with one hydrogen, because we see that the carbon is double bonded. So we see here, carbon, carbon, double bonded with carbon, therefore it only has room for one hydrogen. So we call this a kink. And a kink is a bend in a linear chain. And this kink means that it cannot be fully saturated with hydrogens. If you see any fat that has a kinked appearance, that is going to be a perfect example of an unsaturated fat because it cannot be fully saturated with hydrogens. We see here that the adjacent carbon is going to be similar because it also has 2 bonds with carbon, 1 bond with the carbon next to it, so it only has room for 1 hydrogen as well. So this appearance allows unsaturated fats to be slightly bent, kinked here, and they're going to have only room for 1 hydrogen at this area that is kinked.